Our next speaker is Dr. William Andrews. He has worked in the field of biotech in the industry for 28 years. He has received his PhD in molecular and population genetics at the University of Georgia. He is currently the president and CEO of Sierra Sciences, a biotech company in Reno, Nevada. He has received the second place for National Inventor of the Year in 1997. Presently is named as an inventor on 35 of the United States issued telomerase patents. Welcome, Dr. Andrews.
Thank you. Now, I'm a research scientist. A lot of what I'm going to say right now was just said, so I can probably skip some of it. But I'm a research scientist. The biggest thing I ever work with is a human cell grown in a petri dish. And so you might ask, what's a research scientist like that doing at a conference like this? But I'm going to be talking about the future. And in some cases, the future is now for anti-aging medicine. And but I still got stuck between metabolic syndrome and a cocktail party. So I'm figuring I'm not exactly part of the program. As I was just mentioned, I'm the founder, president, CEO of Sierra Sciences. We're a small company with 30 scientists, and we're completely focused on trying to find a way to induce telomerase to extend our health span and our lifespan. I led the research when I used to work at Geron Corporation that discovered telomerase, and I also led the research where we showed that this could cure cancer and also is a good target to relieve the symptoms of aging. As mentioned, I was awarded second place for National Inventor of the Year. They don't usually award a second place, but that same year was the year that the HIV protease inhibitor was discovered. They got first place, and they gave us second place because we were so close. Telomerase, in my opinion, is going to be one of the biggest things in medical science coming up in the near future in both cancer and aging. So far, my research has covered both areas. I've been very actively involved before Sierra Sciences in trying to find drugs that will inhibit telomerase to cure cancer. But most of my research in the last 10 years has been to focus on finding ways to induce telomerase expression to reduce the symptoms of aging. Let's say I've been in biotech since 1981. I've worked at a lot of biotech companies. I'll skip through this. I've also, because I've been in the beginning, started as a senior scientist right in the beginning days of biotech, I've been fortunate enough to be involved in a lot of very key projects in biotech, including human growth hormone, the development of that, before people thought it would ever be used for aging or muscle building. Tissue plasminogen activator, erythropoietin, et cetera, with the last 16 years of my life being devoted exclusively to telomere biology and telomerase. All right, so let me begin with saying there are a lot of theories on why we age. And I'm a person that really wants to extend my own lifespan, so I'm not going to be biased and say telomeres are it. But what I do is I say all these theories are true. And I like to think of each of these theories as a stick of dynamite that is burning inside of our cells. And so we have multiple sticks of dynamite, one for each different aging process. But it's really only the stick of dynamite with the shortest fuse that we need to worry about. And when we figure out a way to put out that shortest fuse, then we have to figure out how to put out the next shortest fuse. And so those people that have heard the term live long enough, live forever, that's where that came from. We need to keep finding cures for one disease after another. Okay, so my particular niche is to focus on the telomere theory of aging. I'm working on the assumption that it might be the shortest fuse, but I hope that all the other fields are just as actively pursued as my field is. Let me say there's also a lot of theories on how to cure aging or how to extend health span, let's say. Those include stem cells, selection for pristine stem cells. The champion of that is sitting in the audience, Steve Cole's here today. But there's a lot of different things that we can do to extend our lifespans if we want to. Again, my particular niche is control of telomere length, but I still hope that the other approaches are well funded, both in research and development, because I think we're going to need everything done if we're going to be extending human lifespan. But at the same time, I'm very open-minded to what else, what other ideas are there, and I'm an open book. I keep my eyes open. If anybody sees anything that I've left off this list, I appreciate you letting me know. So let me begin at the beginning. Approximately 3.7 to 4.5 billion years ago, the first cell came into existence. And that cell began to divide, and it divided and divided and divided. We are all progeny of that first cell. And during the cell divisions, the daughter cells look like the parents. Now this is getting unwieldy with so many cells, so let's just focus on one line of these cells. And these cells just continued and continued until about 3.6 billion years later. Some of these cells started to differentiate into multicellular organisms, such as cats, dogs, and humans. So now let me name these cells. These green cells, we call germline cells. They're also called reproductive cells. The brown or orange cells are called somatic cells, also called normal cells. And so the question is, why does it appear that the germline cells don't really seem to have an aging process, but as soon as they differentiate into the somatic cells, we start seeing aging? At least aging really didn't exist probably on this planet until 150, maybe 500,000 years ago when cells first started differentiating into adult organisms. So the question is why this is happening. I've been asking that question ever since I was 6 years old and somebody first told me that I wasn't going to be that kid playing in the yard very much my whole life. I was going to grow up and die of old age. And so it's been something I've been very interested in my entire life. My father has always encouraged me to go and become a doctor, cure aging, and that's exactly what I'm trying to do. And I don't know if I can say cure aging, but I mean extend lifespan. So the first example of what I want to show that there is something going on here is that if we take those green cells, the germline cells, we take one of those cells, and a typical example of that is a human embryonic stem cell. If we take those cells, grow them in a petri dish, we find out that they just keep dividing and dividing and dividing. Their daughter cells look just like the parent cells. You don't see any change occur in these cells at all. But if you take a somatic cell and you grow that in a petri dish, at first it grows just like the germline cell, but then after a short while, it starts, after a certain number of days, on the y-axis here is the cell doublings, on the x-axis is the number of days of the cells were grown in culture. But you can start to see that the somatic cells start to slow down and level off in a process called senescence. And in fact, the term for this is called replicative senescence. The more times that as the cell starts to divide, it starts slowing down until it finally senesces. Now, if you take cells from a 5-year-old, skin cells, let's say, from a 5-year-old, it can be any kind of dividing cell, and divide it and grow them in a petri dish, you'll find that they will grow about 60 doublings and then stop. If you take cells from a 90-year-old and grow them in the petri dish, you'll find they only divide about 5 times and then stop growing, and they senesce. This is in intermediate ages have intermediate numbers of doublings. So there's something funny going on here. The cells have something inside of them that tells them how old they are, or how many, let's say, when I say age, I'm usually talking about cellular aging, not organismal aging. So in this case, they know that they only have a certain number of divisions. So there's got to be some kind of counting mechanism existing inside of cells. And this has been known since 1961. This was called replicative senescence. It was discovered by Dr. Leonard Hayflick in 1961. And when you see the cells level off at a certain population doubling, that's known as the Hayflick limit, named after Leonard Hayflick. So the question still is, well, we're answering the question of why the aging. And now we can say that the green cells have no replicative aging, whereas the orange cells do have replicative aging. So something happened to the green cell when it differentiated into the somatic cell, It was inflicted with something. And the question is replicative senescence a disease? And if so, is it something that we can cure? And the question, to begin to answer that question, we've got to first ask, why is replicative senescence occurring? And so that's where the word telomere comes in. And I'm showing it on a newspaper article, newspaper headlines, because this is going to be a very big household word within 5 years. Everybody's going to know what a word telomere is. As you'll see in this talk, telomeres play a very big role in a lot of human diseases, anywhere there's cell division. And for those of you that are trying to follow through the slides, I got to apologize. I made a lot of changes since the thing was printed out, so if you're trying to find where you are in that, I apologize for that. Okay, so telomere is going to be a big household word. And one thing that we've known since the probably 70s and early 80s is that if you take cells from people at different ages and measure the length of their telomeres, you find that the telomeres get shorter. You find a fairly strong correlation, linear line of the older you are, the shorter your telomeres are. So in 1971, Alexei Livnikov postulated that maybe telomere shortening is the cause of replicative senescence, because here we had this counting mechanism. We could observe something that was changing in the cells as the cells divided. And he proposed this in 1971. And so because we're talking about telomeres now, and that's the main focus of my talk, I'd like to summarize a little bit about what telomere biology is. So if we zoom in on a person, you can see that a person is made up, as you all know, of lots of cells. If we zoom in any further, we can see that we can zoom in on the nucleus, where you can see the chromosomes. And in this particular chromosome, I've colored the telomeres yellow. So the telomeres are the very tips of the chromosomes. And now I want to unravel the DNA in that chromosome, in that tip, show it right here. Of course, telomeres and all the chromosomes are made up of DNA as well as protein. But when we are first conceived, our telomeres we can measure our telomeres to be about 15,000 bases in length. When we start to develop, our telomeres shorten, and when we are born, our telomeres have dropped from 15,000 bases to 5,000 bases, or to 10,000 bases. And then when they get to 5,000 bases, we die of old age. Let's just run through that again. So we're conceived at 15,000 bases, we're born at 5,000 bases, and when our telomeres get down to conceived at 15, born at 10, and when they get to 5,000, we die of old age. Now, I haven't shown anything of cause and effect yet. Does aging cause telomere shortening, or does telomere shortening cause aging? One dramatic example of telomere shortening is that some people are born with short telomeres. This is a disease called progeria. A lot of you who have studied progeria know that progeria kids have also been shown to suffer from a mutation to the lamin A gene. But research in the lab has shown that the lamin A gene will actually induce accelerated telomere shortening of cells grown in a petri dish. So even though they have the lamin A gene, which is a protein found in the nuclear membrane of all their cells, this disease appears to be still induced by short telomeres. And these kids is hard to say because they're suffering from all the age-related illnesses that a normal old person would. And their life expectancy is about 20 years. And if there was a way to find a way to prevent telomere shortening, this could possibly be a cure for these kids. So we're back to the question of why. You're going to hear a lot of why today. So we can now say that the green cells don't have telomere shortening and the orange cells do have telomere shortening. And again, something happened when going from the green cell to the orange somatic cell. The cells were inflicted with something. Again, is telomere shortening a disease? And is it something that we can cure? So again, to answer these questions, let's find out a little about why telomere shortening occurs. Most of you that have studied telomeres and read about them in the newspapers have probably heard that telomeres are like the caps on our shoelaces, that when they wear away, they get frayed, and that's exactly what happens to our telomeres. They get frayed away just like the caps in our shoelaces do. Now, I don't like that example, okay? I mention it only because it's so well publicized, but the principal part of telomere shortening has nothing to do with wear and tear. It's a common misconception about telomere biology. What's really going on is a more passive kind of phenomena called the in-replication problem. It's more of a lack of a function than something that's causing the degradation. What I'd like to do is run through DNA replication using an analogy. What I'd like to do is I'd like to compare DNA replication to the making of a brick wall. Okay, so you have look at these bricks here. Imagine that's the top layer of a brick wall. When a bricklayer goes and makes the next layer, that's the same as replicating the DNA. And we know every time our cells divide, our chromosomes have to replicate. So we have a bricklayer that comes along, and he's laying bricks. And remember that our chromosomes are like 10 million bases long, so this is a long, long process. And so the person's got a long ways to go. There's origins of replications about every 10,000 to 100,000 bases. But what we want to really do right here is see what happens when we get to the very end of the chromosome. Okay, so here we scroll over to the telomere, and you can see when he gets here that he's going to have a little bit of a problem when he gets there. And this is exactly what happens inside the cell. He cannot replicate the very end of the DNA, the telomere. And so as a result, the new chromosome is shorter. And this happens cell division after cell division. But no matter what you do, no matter how well you eat, no matter how much you exercise, and no matter how much you do everything your doctor tells you to do, there's absolutely nothing you can do about this. These telomeres are going to shorten like this. Now, I did say that it's not a wear and tear phenomena, but wear and tear, I just said it wasn't principally a wear and tear phenomena. Wear and tear does occur. You can actually accelerate your telomere shortening by leading an unhealthy lifestyle. Examples are anything that would cause stress, and those kind of things are going to cause free radicals, and free radicals can induce damage. And smoking is a good example. Smoking causes free radicals, and that can accelerate your telomere shortening. So the answer is, statement is don't smoke. Don't do anything. And here you can. Exercise helps, eating right helps, and doing whatever your anti-aging doctor says to do, do it, because it will relieve stress. So telomere shortening has been implicated in not just aging, but in a lot of aspects of normal aging. And I'm just going to list a bunch of them and describe them for a few seconds each. Skin aging. We lose skin cells all the time. We're sloughing off skin cells. When those cells are lost, other cells deeper inside, the stem cells particularly, have to divide to replace those cells. As a result, the telomeres get shorter. And so that after many years, your telomeres have gotten very short, your skin cells senesce, and that's why your skin starts looking old. Let's say that's one of the reasons why your skin cells start looking old. Atherosclerosis. We have shown in numerous experiments now that when we take endothelial cells, aligning the blood vessels where there's plaque, the telomeres in those endothelial cells are shorter than endothelial cells found elsewhere in the blood vessels. The same is true for the smooth muscle cells underlying those endothelial cells. But again, especially where you have turbulence in your blood, you're going to be stripping away endothelial cells. Other cells have to divide to replace those cells. As a result, your telomeres get shorter. And when the telomeres get short, cells get unhealthy, and they get a decreased ability to prevent plaque buildup. The common rule of thumb here is bad things happen when telomeres get short. So we want to keep them long. Another one is osteoarthritis. Our joints are covered with chondrocyte cells. Overuse of those joints can cause those chondrocytes to wear away. That's okay at first because you have cells that can divide to replace those cells. But after a while, the telomeres get short, and you can't replace your chondrocyte cells. You get bone on bone. You have osteoarthritis. All these things I'm talking about, if we could find a drug or some treatment that could prevent telomere shortening or reverse telomere shortening, could be a cure for some of these things. Osteoporosis, we have the osteoclasts and the osteoblasts. The osteoclasts are tearing down the bone, the osteoblasts are rebuilding it. Well, the osteoclasts are more dependent on cell division than the osteoclasts. And as a result, telomeres can get shorter. So again, this can result in the osteoblast becoming senescent and lead to the osteoclast dominating, making your bones weak, and is one of the possible causes of osteoporosis. And I got to remember, I'm a research scientist, so most of what I'm talking about is still in the theory stage. We probably won't ever be able to prove some of these things until we actually have a drug that prevents telomere shortening. General immunity. Our immune cells, whenever we have to fight a disease or infection of anything, immune cells have to divide. Telomeres get short. As a result, we eventually suffer from immune senescence. Alzheimer's disease. Alzheimer's disease, could be due to beta amyloid plaques or the tangles, but it's also possible it's due to weakened immune systems. There's a lot of papers suggesting that the telomeres and the astrocytes get shorter in Alzheimer's patients, and it has been shown that just regular blood lymphocytes, the telomeres in those cells get shorter in Alzheimer's patients too. Though there's still no question, nobody knows anything about cause and effect there. Macular degeneration, #1 cause of blindness in the elderly. We have retinopigmental epithelial cells in the back lining of our eyes. When those cells start getting short telomeres, these cells senesce, and that causes macular degeneration. Again, if we could find a drug that prevented telomere shortening, that could possibly be a cure for macular degeneration, if it was the only thing that caused macular degeneration. Liver cirrhosis, I can go on and on, but you're killing your liver cells, other cells have to divide to replace them. Again, telomeres get short. Muscular dystrophy, I can make the same kind of argument. AIDS, the AIDS virus infects the CD4 cells. CD8 cells have to fight unsuccessfully to fight the virus, and eventually the telomeres run out on the CD8 cells, the cells senescent, and people are saying, well, AIDS patients have no T cells. It's because they senesced away from telomere shortening. Not added here is cancer. Cancer is also something that telomere length, even though I've worked a lot on trying to inhibit telomerase to kill cancers, keeping your telomeres long might be a good way to prevent getting cancer. Keeping your immune system strong, and I was saying before that bad things happen when telomeres get short. When your telomeres get short, that causes a lot of rearrangements, mutations in your chromosome, which can cause a lot of mutations that lead to cancer. So keeping telomeres long is also a possible way to decrease the risk of getting cancer. Okay, so other victims of telomere shortening, just research in the lab, scientists working in the lab are always plagued with the fact that they get endothelial cells, let's say, from somebody's blood vessels, and they can only divide a certain number of times, and then they have to get more because they can't do their experiment anymore. So having a drug that they could use would be useful. Monoclonal antibodies, I'm going to skip that one for now. Skin transplants. Well, let's say skin transplants and bone marrow transplants both are plagued with telomere shortening problems. You take the cells out of a person, you expand them in the lab, the telomeres get shorter when you expand them. You put them back in, the cells are older than the person, so at least cellularly older. And the new stem cell technology, that's really going to be plagued with telomere shortening problems, except for the human embryonic stem cells, which don't have a telomere shortening problem. All the rest of the adult stem cells do have telomere shortening. And so whenever anybody is going to take stem cells from a person, expand them, put them back in, just like we were talking about the bone marrow transplants, which is essentially the same thing, we're going to have telomere shortening. So I think the telomere stem cell technology field is going to benefit tremendously from some type of drug that it prevents telomere shortening. This is just a list of all of them. The key thing is there are a lot of them. Learning about telomere biology and controlling telomere biology is going to offer opportunities in lots of different places in the medical field. Any place in the human body where we have cell division is going to be a place where there can be a disease that can be affected by telomeres. So, I would like to say that I believe telomere shortening is a disease, and diseases need to be cured. Okay, so we've said that the green cells don't have any telomere shortening, the orange ones do. Now we can say, let's see, so I guess, okay, I'm sorry. So the question now is, why? So if we want to cure this telomere shortening, we got to first understand why it doesn't happen in our germline cells. And that introduces another word that I suspect will be a household word in 5 to 10 years, and that's telomerase. And telomerase is an enzyme that is like a factory inside of our cells. The gray part of this cartoon is showing the telomerase protein component, the enzyme, and it essentially binds to the very tips of the chromosomes. What's shown in green is the telomeres, the tips of the chromosome. Here now it's shown as a double helix. And the telomerase adds nucleotides to the end of the DNA to lengthen the telomere. So we discovered telomerase on July 6, 1994, and I led this research group. And as we usually do as research scientists, we go out and celebrate with pizza and beer, and had the pizza delivery guy take a picture of us. But the guy in the blue shirt, that's me, and I want to point out 3 other people who I ended up sharing this National Inventor of the Year award with for this discovery. That's Bryant Villapanto on my left in the white shirt, Walter Funk in the purple shirt on the right, and Junlin Fong, who's back in the red shirt. You can barely see her there. But I will be talking about Walter Funk again in a few minutes. Okay, so now, what is going on with telomerase? The bricklayer is still just as unable to replicate the very ends of the chromosomes as he was before. But now, like an angel, telomerase comes in and replaces that missing brick. And so that's essentially, in a concept level, that's what happens inside the cell. So every time the DNA replicates, the chromosomes do not get shorter. How do we know this? When we first discovered telomerase, we couldn't know this before discovering telomerase, but once we did discover telomerase, we did a whole series of experiments at Geron Corporation. As I said before, human cells senesce. Okay, let's say normal human cells senesce. They start, when you put them in a petri dish, they'll divide fine, and then all of a sudden start slowing down, level off, and we have senescence. When we now we have the telomerase gene, now we put the telomerase gene into these cells, and we call this telomerizing them. So when you see the word telomerize cells or telomerizing, that means we put the gene into the cells. It's now a very published term when you talk about telomerase biology. Well, when we did that, we found the cells did not start slowing down, did not have any senescence, and more importantly, they were not cancer. This is like you'd expect a cancer cell to grow like this. These cells were not cancer, and a lot of literature since this time has shown that they actually have a decreased risk of becoming cancer. So this was the absolute first proof. Before I said we didn't know cause and effect. Does aging cause telomere shortening, or does telomere shortening cause aging, or more specifically, organismal aging? We can now say that telomeres are the cause of replicative senescence. So we published this in 1998 in Science. This made headline news, and New York Times called it the fountain of youth. A little premature, but it did make a lot of news. Sometimes this doesn't react fast enough. Okay, we measured the telomeres in these cells, and we did see that they stopped shortening, but they did more than that. They actually got longer. And this was a real surprise to us in 1998, and it begged the question, did the cells actually get younger? And so it was about this time that I left Geron Corporation and went and started my own company, Sierra Sciences. Even though we now are different companies, we still work very close together. But Geron still continues some of the work, and Walter Funk, the guy in the purple shirt that I showed you earlier, did some experiment where he took young and old human cells, and he put the telomerase gene into the old cells, and he also had some old cells that he didn't put the telomerase gene into. And then he cultured these cells into skin on the back of a mouse. Now, you couldn't do this with a human because the FDA would never allow it, but you can do this with mice. So he looked at the young skin, the skin from the young cells growing on the back of the mouse, and they looked young. When he looked at the cells from the skin from the old cells, the skin looked old. But when he looked at the telomerized cells, the old cells that were telomerized, these cells looked young. Well, that was really great, but hold on. You can buy a lot of things in cosmetic stores that'll make your skin look young. So that's nothing new. So Walter took this one step further. He then did DNA array analysis. And DNA array analysis is a procedure where you can look at all the different genes inside of a cell and ask what their expression level is. So he took the cells from the young skin and he looked at it by DNA array. And this picture shows like a computer chip. Each square represents a different gene, and the color represents the expression level of that particular gene. So you can see a profile. This is what's called a young expression profile. When you look at the old skin, you see a very, very different expression profile because a lot of genes are reduced in expression, a lot of genes have increased in expression. But when we looked at the telomerase cells, he saw almost exactly the same expression pattern as the young cells. So for the first time on this planet, somebody had actually made cells younger. And again, I'm talking about cellular aging. So this was very exciting news. It said there was maybe a lot more promise in telomere biology than we originally thought. I'd like to take this one step further. We just looked at skin cells grown in a petri dish, then we looked at skin cells grown on the back of a mouse. Now I want to talk about a whole organism. Now mice don't age like humans do. They don't have telomere shortening. They have telomerase turned on in all their cells. And so it's a really awful animal for using as an animal model when we want to make drugs when we're trying to make drugs that turn on telomerase or prevent telomere shortening. But we were able to engineer, and when I say we, I mean Maria Blasco in Spain, was able to engineer mouse embryonic stem cells so that she destroyed the telomerase gene in those cells, and then she grew those cells into mice by cloning, and then those mice at first acted perfectly normal, because I said mice don't age by telomere shortening. Their shortest stick of dynamite appears to be free radicals, but they're also very driven. Their death is by cancer in a majority of the cases, and predators in the wild. So these mice were developed. After the first generation, they still look normal, but the telomeres got shorter and shorter. Now the babies are born with shorter telomeres than the parents. So eventually, after 7 generations, the mouse telomeres got short enough so that these mice started aging just like humans do. Then she and you see in the bottom corner two other publications took these mice, crossed them into such a way that half the babies would have telomerase, half the babies wouldn't. These mice are the same age, they're from the same litter. The top mouse has short telomeres, the bottom one has long telomeres, and this top mouse is suffering from a lot of age-related illnesses that humans do. Gray and thinning hair, weakened immune system, intestinal atrophy, reduced spleen size, decreased wound healing, a very easy thing to measure in aging, and decreased lifespan. The top mouse lives on the average 2 years, the bottom mouse lives on the average 3 years. Okay, so that was the proofs of principle showing that telomere shortening does cause replicative senescence and cellular aging. And so now in answering this question about why do the green cells appear not to show any cellular aging, the orange ones do, we can say that telomerase, the green cells express telomerase, and that's a typical example of that is the human embryonic stem cell, which expresses a lot of telomerase and has no telomere shortening. And the somatic cells don't have any telomerase. Okay, again, why? Well, even though those cells, the somatic cells, didn't express telomerase, they still have the gene for telomerase. And the gene in the germline cells is producing telomerase. Remember the gray factory thing that I showed you before? This is showing the gene producing the telomerase protein. Now, in the case of a somatic cell, that repressor is, there's a repressor protein that binds to a repressor site, in theory, and shuts off this gene. And so what my company and Geron Corporation spent a lot of time doing is we looked for an anti-repressor that would bind to that repressor protein, thus preventing the repressor protein from binding the repressor site, thus turning the telomerase gene on and lengthening our telomeres. Well, the very first chemical that was ever discovered that would actually do this just came a few years ago, and it's called TA65. And this is a nutraceutical, and it's distributed by TA Sciences, and it's available for research. And they've provided my company with a lot of this nutraceutical so that we could do some research on it and study it. Remember, I'm a research scientist. And so some of the experiments that we wanted to do was, one, validate, does TA65 really turn on telomerase? And we want to do a few other things. But to validate, to measure whether or not telomerase activity exists in a cell, we did a procedure called the TRAP assay. And to explain that, I just want to go through what a telomerase assay is, how it works. Okay, so you take a DNA, a synthetic telomere, and that's what this green thing is here. Telomeres are made up of a repeated unit TTA, GGG, TTA, GGG. And this just has that repeat in there 3 times. If you mix this synthetic telomere with an extract of cells that contains telomerase, such as, hopefully, cells treated with TA65, telomerase will bind, add 6 bases, pause, move over, add 6 bases, pause, move over, add 6 bases. And that's how telomerase works. And it's the only enzyme on the planet that works that way. So hopefully all of you are familiar with electrophoresis, and I won't have to explain this, but if you take those synthetic telomeres, the lengthened one, you'll find that they actually form a 6-base ladder. And if you see that darkest 6-base ladder, that's actually a positive control that we use in our experiments. It's a cancer cell called HeLa, and HeLa constitutively expresses telomerase. So we can see that telomerase activity by seeing that ladder. Now, the next lane over is just showing the same cells, but using 1/20th as many cells, so the ladder is a little fainter. But now the next 2 lanes shows a skin fibroblast, a human skin fibroblast cell line that's not cancer, that's very normal, and it's just as many cells were put into that lane as the 100% HeLa, and there we see no ladder at all, and that's because these cells don't express telomerase. Okay, so the goal of Geron Corporation and my company was to find some way of turning those cells into cells that would produce the ladder, but at the same time not being cancer. And so we've been for years testing everything that came along. Every time somebody published saying that they have some idea how to turn on telomerase, we would test it. Always negative, always negative. Every time somebody would call us up saying they found something that turns on telomerase, we tried it. It was always negative. Try TA65, and lo and behold, we see ladders. Now, I'm going to use a pointer now. You can see the ladders are faint, but they're very visible. They're faint and partially visible, but they are really there. So TA65 is the very first time we've ever seen something that actually induced telomerase expression. And as you can see in the lanes that don't get the drug, there is no ladder at all. Well, it was faint enough that we thought, well, maybe this is just some artifact, maybe it just got some weird thing happening that day. So we repeated the experiment, and again we see it. Now in this case, we can see a little more dose dependency. The higher doses are showing more of a ladder than the lower doses, which is good. So we repeated again, and again we get this. So we repeated this several times, and we very reproducibly can get TA65 to turn on telomerase in normal cells. And it's the first nutraceutical, chemical, anything on the planet that can do this. As if there was something, well, let's say there's a few other ones now, but I'll talk about those later. TA65 was also shown not to be toxic, at least by the experiments that we've done at Sierra Sciences. One is to just, we test a lot of drugs at Sierra Sciences, and we always see toxicity to some degree or another. And this particular nutraceutical was showing practically no toxicity whatsoever, even at the highest doses. So if you look on the right-hand side of the slide, you'll see cells treated with nothing except DMSO, and then the other cells treated with TA65. And I don't know how clear you can see it, but you can see that the cells look exactly the same and about the same density. We did a lot of other tests for toxicity, including just cell counts in a culture counter, cell titer glow, which is measuring ATP levels, and PsyQuant, which is measuring nucleic acids in the cell. And in all cases, we saw no or very low toxicity. Another thing that we did to kind of look at toxicity is we did DNA array. Remember I talked about DNA array before in that human skin grown on the back of a mouse model. In this case, we took cells treated with TA65 and then asked what happened to the expression pattern. So we had cells that were untreated and then cells treated with TA65 and asked what kind of differences did we see. This graph is a little hard to explain without a pointer, but let me try it. The x-axis is just showing the full difference in expression. So if you see that number 32, that says 32 different genes showed a twofold decrease in expression levels. And then 9 genes showed a twofold increase in expression level. Well, that's practically no change at all, if anybody who's used to looking at DNA array results. And what I want to show is that Sierra Sciences is trying to develop drugs too, and we've actually now discovered about 120 drugs that turn on telomerase, and we're trying to develop those, but none of them work as well as TA65 yet. And this is our best case of doing DNA array analysis with our best drug, C00576684. And if you analyze this data a lot, you would see that about 10% of all the genes in the entire genome are affected at least twofold in their expression level. So TA65 is about the most inert chemical that I've ever seen in a DNA array analysis study. So TA65 is a nutraceutical extracted from the traditional Chinese medicine astragalus root. TA Sciences did some studies themselves, and the first study was to test 36 men ages 60 to 85 in a 24-week study. So this is the results of the study. They looked at immune system improvement, vision improvement, skin improvement, sex improvement. Now, even though you can see there's a trend, but a trend that the blue bars are treated with TA65, the gray bars are not treated with people that didn't get TA65. And you can see that there's a trend that people who treated with TA65 were showing improvement in a lot of these tests here. Now, if you look closely, you'll see the statistical scores aren't that great. There are statistical scores like p.12, p equals 0.47. So the sample size was too small. But look at this score here. This is the immune system, lymphocyte proliferation. There was a improvement in lymphocyte proliferation in patients taking TA65 that was significant to the 0.005 level. Now, a lot of people, especially me, believe that the #1 cause of aging is a weakened immune system. And so it's good to see that TA65 is having such a dramatic effect on the immune system. Now, there's a lot more data on this. I'm not going to go through all of this because especially since it was a small pilot study, but you can see the entire study at www.tasciences.com. Now, in the last 2 years, a lot of people have been taking TA65, and that includes, I'm one of the people that's taking TA65. I've been taking the drug myself for 2 years. And a lot of these people that have been on for more than a year, finally we're offering some data. We've been getting our blood samples taken like every 3 to 6 months, tons of biomarker studies. You had to agree to all that in order to be able to purchase TA65. And after looking at a lot of the clients that have been on it for 1 year, definitely seeing some good things happening here. CD28 negative T cells. I'm not exactly certain what CD28 negative T cells are, except they're T cells, and they've been heavily published in the literature to be very correlated with aging. When you age, the older you get, the more CD28 negative cells you get. But the clients taking TA65 have shown a very statistically significant decrease in the CD28 negative T cells. Naive T cells have gone up, natural killer cells have gone down, blood pressure has gone down, skin elasticity has gone up. All these things correlated to what you'd expect if TA65 was actually helping reduce the symptoms of aging. So personally, I'm an ultra runner. I run 50, 100-mile races a lot. I've been doing this for 13 years. This is showing me running the Western States 100-mile race. This is running the Badwater 135-mile race in Death Valley. I've always been back of the pack runner. And in a lot of my races, I finished dead last. And you know, I showed data where I showed that TA65 is causing telomerase to be produced inside of our cells. Well, from all that data, I would have expected that TA65 was very weak, couldn't possibly be doing anything that would improve anything like my running. But only 3 weeks after I got onto TA65, I started finding that my running performance improved dramatically. I started finding I was running past runners that beat me by hours every time. I was going like this, saying, I don't know what's going on. I ended up in that 3 weeks after taking TA65, I finished 9th place overall, first in my age group out of over 100 runners competing in the thing. And I couldn't figure out anything else except TA65 because nothing else has changed. And it's been like that ever since. I've been on TA65 for 2 years, and it's like, I don't want to go off it. I'm having such a great time with it. So I'm going to finish off with, does telomere shortening cause aging? And we don't know yet. We don't know. We can say it causes cellular aging, but nobody's ever been able to show that cellular aging causes organismal aging. And the only way we're ever going to show this is to develop a drug that does reverse telomere shortening, re-lengthen telomere shortening, at least stop the shortening, and show that it affects aging. TA65 might be such a nutraceutical, but it's going to take a few years to actually find that out. Is aging a disease? Again, we don't know if aging is a disease. The consensus among a lot of people is that it's not a disease, but Sierra Sciences and Geron Corporation are trying to cure aging, and if we succeed, then then it says that aging is a disease. So, any questions?
I just don't understand in your graphic.
I can't hear you.
In the graphic where you show that the telomere is able to allow those building blocks to be put back on, I just don't understand genetically how is that possible if in fact that's now a shorter. Where's the code? As I remember from medical school, you need to have it to then to copy itself. So what's the blueprint? How do we know that what's going to be built is in fact appropriate?
Okay, maybe I don't understand the question, but the telomeres are made up of TTAGGG. They don't encode anything. There's no genes there.
Okay.
And so you've got like 20,000 or more bases of just these T2AG3s. They're there just to stabilize the chromosomes, to protect the chromosomes from rearrangements. And when they get shortened, they really don't change anything in the gene sequence. And
okay.
In a given individual, is the telomere length the same in all cells, or do some?
It depends on the number of times a cell has divided. Okay, so when you're born, your cells, when you're a single cell embryo, you have a certain length. Well, as they start dividing, some are going to divide more than others. Those ones that divide more are going to have shorter telomeres. Plus, there's a statistical Poisson distribution because remember, whenever there's a short telomere, the long one still is around. Okay, so there's going to be a distribution, random distribution. And right now, it's looking like it's really not the average telomere length that's important. It's the number of short telomeres you have.
I guess what I'm getting at is there any way to test in a given individual what their physiologic age is by measuring telomere length?
That happens every day right now. TA Sciences does that with all their clients. SpectraCell is a company that's going to start offering this test in June for $300 per patient.
Using what cells?
They're going to, well, they'll be using the blood blood cells, but they could do it with any cells if you can provide a way to provide it to them. There's been lots of papers published on looking at every cell from endothelial cells to a host of cells. I can't remember them all, but it's in all cases of dividing cells, telomeres get shorter. But when you look at different tissues, you do see different telomere lengths.
Thank you.
Any other questions?
When you're looking at increasing or allowing cells to continue to divide, what mechanism is in place when you're looking, I guess the thing that came to mind, how do we know that you're going to take this nutraceutical and not induce some kind of increased cell multiplication where you're going to induce a cancer?
Okay. So there is 85 to 95% of all cancers express telomerase. But telomerase has never been shown to cause cancer. In fact, telomerase has been shown to decrease the risk of cancer. Nobody's ever found a cell, a human cell, that expressed telomerase naturally that wasn't cancer. Cancer always has to come first. And it appears that one major way to cause cancer is to let the telomeres get short. And that might be one of the reasons why cancer is more prevalent in the elderly. But the rest of your question would be, it would be like any other drug or nutraceutical or anything like that. It has to go through some testing, and that's why we did things like DNA array analysis, because if it was going to cause cancer, we'd probably see some change there. And also, all the clients that are taking TA65, I'm not privy to all the data from there, but I do know that nobody has seen any adverse effects whatsoever.
So there haven't been any adverse effects? Is that what you said?
Right.
Okay, thank you.
I have a question. How do we get the TA65, or how do we learn about studies regarding this?
TA Sciences is here. They have a booth next door. But Jeff Life, he was here a few minutes ago. He is also somebody you can get TA65 from. But it is available. And but since I'm not, and there's Greta Blackburn walking in the back right now. She's somebody you can talk to about that. Okay.
One more question. Is there anything else that increases or vitamins, minerals that maybe work with TA65 or in conjunction with? Sometimes we're putting different things together would make this work a little bit better, or?
Nothing so far, because really all the metabolic pathways that occur inside of a cell really have nothing to do with the telomeres. The telomeres affect them as opposed to the other way around. I did say that we have now found 120 different drugs that turn on telomerase. They all have a little bit of toxicity, but we plan on doing synergy experiments, mixing them together to see if there's some way to improve the potency. And TA Sciences has agreed to let us do it with TA65 too. So
thanks.
Okay.
Stephen Coles from UCLA. As you were talking, I started thinking about the area of my hobby, which is the stem cell arena. And there are two basic approaches now that are being discussed about how to create a large or amplified set of cells that would be autologous and not rejected if we infuse them back into the body. One is a method that uses embryonic stem cells, which is politically very controversial. But the new method, discovered originally, I guess, in Japan, and now, the work reproduced not only at Wisconsin but at UCLA and many other labs, is called induced pluripotent stem cells. And a lot of people say that iPS cells are equivalent to embryonic stem cells. So all we need are adult cells from a skin biopsy. We don't need to do embryonic stem cells. That's what the religious conservatives are telling us. And it just occurred to me that if you derive an iPS cell from an adult cell, how would you have a telomere length that was equivalent to an embryonic cell? You wouldn't.
Right.
And if aging is related to telomere length, we may be giving cells which are based on iPS cells that are old instead of young. Does that make sense?
Yes, it's very likely that iPS cells will need a telomere extending drug in order to make them really effective. We are working with iPS cells at Sierra Sciences, and I don't know if you heard just last week or something, they've now shown that you don't have to put the 4 genes into the cells. You can actually just bathe them in the proteins. So that's going to, that's going to make everybody, it's going to make it so we all can have our own human embryonic stem cells without sacrificing an embryo. Any other questions? All right, so I think it's the cocktail party.